right? And Mendel and even Darwin and everybody else saw that not everything was the same. So they didn't know what the heck was going on. Now keep in mind, Mendel had no idea. Nucleus, no idea. Chromosomes, DNA, nothing. So all he did was look at traits and analyze probability, and then he published his papers. Now initially he went to work with mice. Luckily he didn't. That was a disaster. So he wanted the head abbey guy and goes, well, I'm going to do heredity studies with mice. And the head abbey guy goes, well, you know, mice having sex, we're still a monastery, that's too sexual. But this is 1700, 1800, not my ticket, right? So he said, no, you need to talk. You have a greenhouse, and no one is using it. I want you to do your experiments over there. And that was it, because what Mendel did, he started working on the garden pea plant. Garden pea plant. He selected garden pea plant, which is an excellent choice. And the scientific name is Pisum sativum. Notice genus name on the line. So on the exam, if I ask you who's the father of modern genetics, the response would be Gregor Mendel, a monk, Father Mendel. And what was he working on? Garden pea plant, also known as Pisum. Now, I wish I had my transparency, but we don't have a machine here for me to project the machine. But with the Pisum sativum, what's interesting, the male and the female part of the plant stands out really well. Remember the stamen and the carpel? They really stand out, so you can do cross-pollination. So you can select for certain traits and do cross-pollination. You put them in and look at the offspring over time, right? So you start doing that work, and based on his work, he laid the foundation of modern genetics. Now, unfortunately, he published it in an obscure journal, and that stuff kind of disappeared. No one read it until 1900. So 3 people discovered it at the same time, accidentally, 3 of them. And that was the beginning of the genetic revolution. 1800s to 1900s, 1870s. To 1900 when it was discovered. From 1900 to 1950 when DNA was actually discovered. And then from that point on, now we can use from one animal into Alright. So, let's talk about Mendel, what he did, everything else. Again, it's amazing that the amount of work he did because he had no clue what genes were. He had no clue what DNA was. So imagine like you taking an exam last week without knowing what chemistry is, what redox reactions are, what energy carriers are, what bombs are, nothing. That's not even genetic research. It's going to be very difficult, isn't it? So it was amazing stuff. But before we talk about him, let's talk about terminology. Because you need to understand the language of genetics. If you understand the language of genetics, you're going to do well in this next exam, okay? You're going to do well. So it's the foundations of genetics. The first thing we're going to talk about, the first term is called the gene. How many of you heard of this term? Right. Now, why don't you go ahead, look around you. Look around the person next to you, look at the person behind you. Go ahead. Look around, go over, look around. Go ahead. Right? You ever wondered why some people are ugly and some people are not? It's genetics, right? And that dilemma will be solved today. That dilemma will be solved today. Genes are nothing more than... Let's do another thing too. Here, hold your hands like this. Hold it like you're being facing me. And then clasp them together. Now, how many of you have your left thumb on top? How many of you have your right thumb on top? Now try going the other way. You actually have to think about it, right? That's actually genetically determined. Now, this is called the pleiotropic effect. It's not like we have a gene just for this. There is no evolutionary significance. Like the ones who trade this way versus this way survive, right? There's a gene for another trait that influences this. That's called pleiotropic multiple effect. But that has also been determined by genes. Genes play a big role in our lives. Even in our behavior. Believe it or not, we have genetic predisposition for certain behaviors. Right? Criminal behavior, altruistic behavior, there is genetic basis to it. But what happens is just because you have that gene doesn't make you a criminal. You're environment also plays a role. Environment plays a role. How many of you heard of Iceman? Probably the most ferocious serial killer. He used to kill for a mom ever, all right? This guy has a predisposition for that. No apathy at all. But he grew up in a very conducive environment. There's other people who carry the same genetic problems, but they grow up in a loving environment and never manifest itself. But this guy, it does. I remember I was watching them on HBO, they had a special, they were interviewing him, and the psychologist asked him a question, and he kind of got annoyed and gave him this look, and I wish I could practice that positive. I was trying to practice that. I was scared, and I was sitting at home on the couch with that look that he gave the psychologist guy. Like an amazing look, but he had that apathy in it, which is pretty amazing. It's pretty scary, actually. All right? So genetics play an important role, but environment also does. So don't think it's all genes. Because if gene makes you criminal, then you're not liable for your crime, are you? We know it is the behavior. But clasping your hands, rolling your tongue, being ugly, being not ugly, all of that is turned by. So what are genes? Genes are nothing more than units of information about a specific These are nothing more than units of information about a specific, and they are found on chromosomes, right? Found on chromosomes, DNA. And then their location is called the locus. So what is their location called? Locus. So genes are units of information about a specific trait. They're found on the chromosome. Their location is called locus. There's a multiple location called loci. All right. But if I ask you what is a locus, your response is where genes are located on the chromosome. That's the address of the gene. Now, we have genes for hair color, eye color, etc., right? But genes come in different molecular forms. So genes come in different molecular forms, and this is what we call alleles. Alleles are genes. Allele and gene mean exactly the same thing. But since genes come in different molecular forms, we tend to call them alleles. All right? So don't get confused on the exam. If I say, what's the location of an allele on the chromosome called? You still say? Because alleles are genes. It's just they come in different molecular forms. Because genes could be either dominant, or I should say, allele could either be dominant, or it could be recessive. Generally speaking, a dominant allele is shown with a capital letter, and recessive allele is showing with a lowercase. Now, this is important. In a classical Mendelian sense, in a straightforward Mendelian sense, okay, dominant allele always wins out. Make a note of that. In a classical Mendelian sense, which one wins out? Dominant. On the exam, we have exceptions, and I'll tell you if there's an exception. You don't have to kind of, oh, is it classical or non-classical, okay? I'll tell you if it's an exception too. But generally speaking, the dominant allele will win out. If you have a gene or allele for a dominant eye color, and allele for a recessive eye color, which one will win out? The dominant. Okay? The dominant always wins out. Now we'll talk about specifics later, all right? So any questions on genes and alleles and everything else? That's the biggest part. Now, and I apologize for this word, I have to constantly wait. Humans are diploid, right? All organisms are diploid, right? Diploid means what?
Two half cells.
If I say you're such a diploid, am I catching you out? It's a diploid. What is diploid? That means your chromosomes come in, right? Your chromosomes come in You inherit one set of chromosomes from mama and another set of chromosomes from, right? So you get that and, sorry about that. So, if alleles, can be either dominant or recessive, right? There are different possible combinations. You can get two dominant alleles, right? One from mom, one from dad, right? You can get a dominant allele and a recessive allele, right? Or you can get two recessive alleles. Is that true? These are the possible combinations. Now, we need to know what these mean. If you have two dominant alleles, this is called homozygous dominant. So if you have two dominant alleles, it's called homozygosity stands for dominance. Homo means same. Homozygous dominant. If you have one dominant, one recessive, we call this heterozygous. Heterozygous, you have two different ones, right? And if they're both recessive, you have homozygous. You need to understand, because on the exam I'll give you word problems, and from those word problems you have to be able to calculate probabilities. Well, if you don't know what this means, you're going to be trouble. So, homozygous dominant, you have two dominant alleles. Everybody with me? Heterozygous is one dominant and one, and then homozygous recessive is both of them happen to be extremely important. Now, this homozygous dominant, heterozygous, homozygous recessive, all of this stuff, all of this stuff, is called a genotype. Genotype is nothing more than the genetic makeup for a specific trait. Okay? So your genotype is nothing more than what? Genetic makeup for a specific trait. Everybody with me? But you also have another concept that you need to know, and that concept is called phenotype. So a genotype is a genetic makeup for a specific trait. Phenotype is nothing more than what we call observed traits. Phenotype is what? Physical characteristics, right? Technical definition, it's called the express trait. This is a trait that is expressing itself based on a specific genotype, observed trait, based on a specific genotype. Karina has brown hair. That is her phenotype. Okay? Brown hair is what? Phenotype. Right? You have brown eyes. That's an example of phenotype. Left thumb on top or right thumb on top, that's an example of phenotype. Now, what's causing that phenotype is called a genetic basis, is called a genotype. Now, we'll talk about what are the possible combinations. Which one can give you one. But I want you to understand what is the genotype and phenotype. So an example, if I say a specific organism with this kind of a trait, automatically you know I'm talking about phenotype. One mate with this individual, how many dominant alleles they have and so forth. You have to be able to take this, translate it into this, and then do your calculations. Right? So, now, remember I said dominant trait always wins out? So if your homozygous dominant, do you think you have a dominant phenotype or a recessive phenotype?
Dominant.
Dominant phenotype. What if you're heterozygous? Right? As long as you have one dominant allele, this is different forms. As long as you have one dominant allele, you will get a dominant phenotype. All right? So again, you have a and then if you are homozygous recessive, then what kind of a trait do you have? I should just use trait. Same thing, trait. So those of you who have blue eyes, true blue eyes, not hazel, is dominant. It's true blue eyes, true blonde. That is a recessive genotype. And since a recessive genotype, that means you are definitely homozygous recessive. That means you are homozygous recessive. Everybody with me? Okay. You are homozygous recessive. That's the only way you have a recessive trait. From the classical Mendelian sense, you're recessive on both alleles. But as long as you have one dominant allele, you have a dominant trait. Now, I have black hair, well, white now, because of marriage and having kids and you guys. That's my phenotype, right? But can you tell by just looking at me, am I homozygous dominant or heterozygous? You can't, right? So I can be either or. On the exam, if I say an individual has a dominant trait, look at it. Beautiful brown eyes, you melt when you look at them. That's his phenotype, right? So what are his possible phenotypes? Homozygous dominant or heterozygous? You select both choices, right? Because there's no way for you to. Everybody with me? All right, now I'll talk about how you can determine. There's breeding experiments you can do. We can't do it with humans. Because in order to get a proper number with humans, the woman has to give birth to 50 babies. That is statistically viable number, and you can do your analysis. Now, what woman will have 50 babies? If any man goes up to a woman, I want to marry you, but I want 50 babies. I don't care how much he's in love with him, she will walk out faster before he what? Right? Okay? Which is, it should be. No one should have 50 babies. My grandma had 12, but that was way back in dinosaurs. Any questions so far? Okay. So what is the locus again?
It's a
Location of an allele on a
gene.
chromosome. Alleles are nothing more than different forms of genes, which are the same. So you can technically say alleles are genetic units of information about a specific trait, but they come in two different forms, dominant or recessive. And of course, if you have two dominant or one dominant, one recessive, you call it a dominant phenotype. So if you have recessive, you will have recessive. And that's what these terms mean, all right? It's called the genotype and the phenotype. Pretty much the physical characteristics, the smell, the look, etc., etc., etc. All right? Then there is a concept, and this might get a bit iffy here, but bear with me because this has direct implications on Mendel's work. Something called a true breeding lineage. Okay? This is directly informed from Mendel. So true breeding lineage. What is lineage? Your ancestry, right? A true breeding lineage is pretty much offspring of crosses. By the way, every time you hear the term crossing, that means mating. Okay? So in genetics, when I say crossing, it's the same as cross-pollinating. Plants mate, right? Humans mate, animals mate. That's what we call crosses. My son is a product of me and my wife crossing. You are a product of your parents crossing, right? Crossing, that's the term we use, okay? It's not this crossing, okay? There's essay components, and sometimes you get confused, and they say, well, we cross ourselves in church. No, different crossing, all right? True breeding lineage are offspring of genetic crosses that inherit a pair of identical chromosomes. That a pair of identical chromosomes. Now, I'm never going to ask you to define it. You just need to understand. Simply put, let me put it in common English. Simple English. True breeding lineage are when you're crossing two homozygous recessive individuals together, or when you're crossing two homozygous dominant individuals together. Got our first? So if you're crossing, when you're crossing two homozygous recessive individuals with each other, you're always going to get homozygous recessive offspring, right? And if you're crossing two homozygous dominant individuals together, you're always going to get what kind of a trait? Homozygous dominant, or just a dominant trait. So that's what true breeding lineages are. Because what Mendel did, he took a true breeding lineage for a recessive trait. How do you know it's recessive? Because he crossed it millions of times. And you knew this is all you get, all right? Established that trait, and then he crossed it another group, and he formed that group, and then he started cross-pollinating and looking at ratios, okay? But a true breeding lineage will always give you that either dominant or recessive. There is no heterozygous involved, right? You just focus on here and there. Everybody with me? Any questions? Allele, that, breeding lineage, locus allele, homozygous dominant, we covered that. And then, of course, you're going to see stuff like this. P stands for the parent generation. F1 is the first generation. So your parents are P, right? You are F1. If you have children, they are F2. If children are F3, and so forth.
Professor, what is that called? Is that still part of the true breeding lineage?
Are you talking to yourself, or are you asking a question?
I'm asking a question. Is the P1, is that part of the true breeding lineage?
Well, no, just P. There's no P1. It could be a true breeding lineage, or it could be anything.
Anything?
Okay. Now, Mendel selected true breeding lineage for his experiment, but is any one of us true breeding lineage here? No, it's very difficult because our genomes are so complex, right?
Yes.
We don't have that true breeding lineage. But with plants, you have certain plants that always have red flowers. You have certain plants that, if you cross two reds, you always get red. Or if you cross true whites, you always get white. Those are true breeding lineages, right? So much easier if you know with that.
Pure breeding.
Okay? All right. So, let's talk about Mendel and what he did now. Let's talk about Mendel and what did he do. Any other questions? Ask questions. Right.
The homozygous recessive is also related to people with blue eyes?
Well, if blue eye is a recessive trait, right? So that means they are homozygous recessive for that trait.
So people with brown hair can have that one?
Brown hair?
Yeah.
Well, brown hair is a different allele. Blue eyes is a different gene, right? You have a gene for your eyes and you have genes for your hair. Brown hair is a dominant one. It could be either this one or this one. You never know. Okay? And after we do Mendel stuff, we're actually going to play with numbers. Okay? So let's take a look at Mendel. Any more questions? Remember, asking questions is good. That's why you guys are lucky in the class. This is asking questions. The rest of them do. It doesn't matter, top 35. So let's take a look at Mendel. Let's take a look at Mendel. Right? Take a look at Mendel. Is it M-E-N-D-E-L? Mendeli. If you know him as well as I do, you call him Mendel. By the way, Thanksgiving is coming up. If you want to read an excellent book about Mendel's life, it's called Monk in the Garden. It's a biography of Mendel. And I even have a copy of it. If you want, I've lent it to you as long as you bring it back from perfect condition. Because my books are sacred for me. I'll lend it to you to read. It's an excellent short book, 140 books. You can read it over.
140
Yeah, sure. It's like one weekend read. It's nothing technical. But Mendel did crosses, and the first set of crosses he did were monohybrid crosses. Monohybrid crosses. What a monohybrid cross is, that means you are crossing for one trait. So you're focusing only on one trait, right? Red flower versus white flower, all right? Wrinkle seed versus non-wrinkle seed. Tall plant versus non-tall, which they call dwarfing plants, right? So you only focus on one specific Everybody with me? Now, which one is my transparent? Let's see, which one do you use? I'm going to use the white, in this case it was a purple flower. You know what? I'm going to use another example this year. Tall versus dwarf. Tall versus dwarf. So what he did, or his T1, this is amazing. He's just looking at traits. And by the way, he was doing thousands of crosses. You can't just do one cross and look at numbers. Now, you look at a couple and look at their baby. And let's say the couple has brown eyes and the baby has brown eyes. You can't still tell if the parents are homozygous, dominant, or heterozygous. Because maybe the second baby will have blue eyes. Okay, we'll talk about those probabilities later. That's why a woman has to have 50 babies to be statistically viable. But these guys is plants. He did thousands of crosses and he looked at ratios. So for the parent generation, he crossed a true breeding lineage that happens to be tall with a true breeding lineage that happens to be. So he crossed a true breeding lineage. I'm not writing it down, but he crossed tall with small. And then he looked at the offspring, and he found in the first generation all the offsprings were tall. So he took a true breeding lineage, everybody with me? Tall. True breeding lineage, dwarf, and he crossed them, and all the offspring, all the plants, thousands of them, they were what? They were tall. So based on this, Mendel said, so which trait do you think is dominant?
Tall.
Everybody sees that, right? Tall is dominant, always went down. So he said, he started assigning codes. So he said, since this is tall, he defined in the code capital letter T. Right? 2 capital letter T. Everybody with me? And since this is a dwarf, you sign the lowercase. Now, you don't put D. What you do is lowercase t, right? You do lowercase t. See what happened? Because that's telling me this is dwarfness in relationship to this is extremely important for you to know. Anything slightly missing, raise your hand. Everybody getting it? Okay. Now, all offsprings are tall. Well, what is their genotype? This is what he did. He did what we call Punnett square analysis. You need to understand Punnett square. You develop a square, like this, Punnett square, 4 squares in it. And let's say, hypothetically, this is the male, right? Well, when the sperms are diploid, they come in pair, right? Haploid, they split in half, right? So you split those 2 in half, put them there, right? You do the same thing here. You split that one and you put it. That doesn't matter if you put it here, or put these on top, these on the bottom. Doesn't matter, okay? Now, what are the possible combinations? Well, this sperm can combine with this egg, right? This can combine with this one, right? I'll do arrows for the first one. This can combine with, and by the way, generally speaking, you always put the dominant first. This can combine with this one, right? So notice, what is this genotype called? Homozygous dominant, heterozygous, or homozygous recessive?
Dominant.
So all are heterozygous, right? All heterozygous. One dominant and one recessive. Everyone with me? Because, take a look at the genotype. Is this tall or dwarf? Tall or dwarf?
Tall. It's dominant. Tall.
And that's what he was getting. Everybody with me? Now he's not done yet. Then he did an F2 cross. And what an F2 cross is, he took a bunch of F1s and crossed them with each other. He took the F1s, so in other words, he took F1 and crossed it with another F1. Right? Now, what is the genotype for an F1? Actually, let's go. What's the genotype for an F1?
Tall.
He crossed it with tall. Right? And what are the genotypes? What were the genotypes for the F1?
FT.
Heterozygous. Heterozygous. Crossing 2 heterozygous. And Mendel said, if this is correct, if my analysis is correct, he said, if I'm correct, I should get a ratio of 3 to 1. 3 tall to 1 dwarf. And I was tempted to say short. All right? So 3 to 1. Everybody with me? Now, let's do the Punnett square for that one, for the F2 crosses. Again, you draw your Punnett square. You have one dominant and one recessive, right? Pay attention to me, please. You have one dominant and one recessive, right? Dominant, recessive. And then you have one dominant and one recessive. Everybody knows how to do this now, right? Well, this and this will combine, it will give you this, right? This and this will combine. This and this will combine. This and this will combine. Now, let's take a look at their phenotype. Tall or dwarf?
Tall. Dwarf.
What are the ratios? By golly, Miss Mo, and there was not. So when you cross those, right, when you cross the offspring, you got that 3, you got a 3 to 1. Genius! He had no clue what genes were. He had no clue what chromosomes were. He just looked at traits, assigned them code, and did the probability. Now, you think he's done? No. Because we have a 3 to 1 ratio, tall to dwarf, right? Well, how did he know which tall is homozygous dominant and which tall is heterozygous? He did, right? Did he have the genetic tools to do a gel electrophoresis? By the way, good news, we are reviving our biotechnology course in this campus. We'll do genetic fingerprinting, all that CSI stuff, genetic fingerprinting, DNA escalation, all that kind of stuff. So if you want to get into the criminal justice maybe, or stuff like that, or if you're interested in biotech and manipulating genes and stuff like that, that'd be great. I'm going to take, as soon as it's offered, I'll be your classmate. All right? We can study together. I can cheat off their hands, right? So, in order to determine which one is tall, and I've taken courses before, so it's fun. And students try to compete. Even though, I'll be honest, some of you at this level, are much better students than I was at this level. I'm being honest. My first semester in college, I went on probation. Because all I focused on was party, soccer, once in a while I stayed. And I went on probation. And that was a wake-up call. I'm glad it was the first semester, and I was able to eventually get my doctorate. If you have a rough start, it's not bad. You learn from it. But if you constantly have a rough start, then, like Taylor Swift, how many breakups she had? Eventually she needs to write a song that's all about me, or something like that, or maybe it's me. So, I'm just saying. Now, in order to determine which ones were tall, which ones were dwarf, what Mendel did is what's called a test box. Because his talls are unknown, right? The tall is unknown. Everybody with me on that one? Based on that Punnett square, the tall is either this or this, right? Right? Imagine, he has thousands of these plants that are tall. Remember, I think the actual number is 268,000. No, 2,068 or something like that, 680, something like that plants. Tall or dwarf. Now he wants to see which one is dominant, which one is heterozygous, to actually test it. Well, a test cross is when you take an individual, you take your unknown, and you cross it with a true breathing lineage. You take your unknown, and you cross it with a true breathing lineage for a recessive trait. Again, you take your unknown, and you cross it with a true breathing lineage. TBL stands for true breathing lineage, okay? You cross it with a true breathing lineage for a recessive. Now, what do you think a true breathing lineage is when it comes to the height? Is it going to be tall or dwarf? So true breeding lineage here is dwarf. And what's the code for that?
Small t, small t.
2 lowercase t's, right? And use the board. It'd be ideal if I can leave this stuff in there so I can come back to it and refer to it. So this is what has happened. These are the possibilities, okay? Now we're crossing for a true breeding lineage, right? Just like here. This is given, right? And then of course you have a tall one. Now the tall one's going to be either this or, right? So a tall was going to be either this or, everybody following me here? Because I don't want to lose you. Everybody with me? Yeah. Who's not following me? It's okay. Okay.
Why did you make that box with the one box with 2 uppercase?
One box, because these are the tall ones, right? And if you're tall, you're either this or this. So you know, we don't know which one is which, right?
Yeah.
So he's doing all these test for us. Okay? Now, if your tall one is homozygous dominant, he said, well then all my offsprings must be tall when I do a test cross, right? So if I'm crossing this with this, you're always going to get tall offsprings, right? So go ahead and do this cross. So everything comes out to be heterozygous, right? Therefore all tall. So if he did a test cross, he took the tall one and crossed it with a dwarf true breeding lineage, and he always got tall, his conclusion was, well, this is a homozygous dominant. But if your tall one is heterozygous, what you get is a 50-50 ratio. If it's homozygous, if it's heterozygous, you get a 50-50 ratio, because these 2 crosses, this 2 crosses, these 2 cross, this one, that one cross. This is tall, this is tall, this is what? Dwarf, this one is dwarf. Therefore, you're getting a what? 50-50 ratio. So if you do a test cross and you get 50-50 ratio, then say, well, this individual happens to be what? Hetero.
Zygous.
Okay, so you said that most humans don't know when it's like a true breeding lineage.
Because you have to do a lot of crosses.
Okay, so but if my dad had brown eyes, my mom has brown eyes, and I have brown eyes, wouldn't either be a true breeding lineage?
No, because, we'll use that as an example. Let's just use that as an example, okay? Digress a second, if you get me. Let me, your dad, your mom, brown eyes, right? They cross. You are, this P, F1. You are born. You have glasses on your head. They have brown eyes, right? That's a dominant trait. Well, if your parents are homozygous dominant, you always got to be homozygous dominant, right? If you do the Punnett square, you always get big E's, right? But what if your parents are heterozygous? They have a dominant and recessive. What eye color would they have? Still brown, right?
Yeah.
But when you do the crosses, there is 1/4 chance, or 25% chance. There's 25% chance that you will have, but it's a luck of a draw. You happen to be any one of these, right? So in order to be valid, your parents have to have at least 50 kids to completely eliminate the possibility. So if you're really curious, go to grandpa. I want to know. You have any other siblings?
Yes. They both have brown eyes too.
How many kids? 2? So they have 48 more to go. All right? That's 48. Now, chances are very high that they might be, you never know for sure. You never know for sure. Okay? You never know for sure. Now, if there's a history of brown eye in your family, your parents might be carriers. Right? So it is possible that you have heterozygous trait. And if you marry somebody who has blue eyes, all right, or he's heterozygous, there's a chance that you will have a baby that has blue eyes. It's very possible. All right? Does that answer the question?
Yeah.
So you're not true breeding lineage, you don't know.
Oh.
48 more babies will know. But at least in respect to your parents, right? Yes.
How do you determine what's dominant, like between brown eyes and dominant blue eyes?
Because, well, brown always wins out over, no, if you have somebody's brown eyes, mates with somebody's blue eyes, and if that brown eye happens to be, for example, dominant for sure, they're always gonna give you brown eyes. Okay? Green eyes is still considered a shade of brown. You either true blue or everything else is just different shades. By the way, eyes are a little bit tricky too. It's not as simple as we're talking about. There's a couple other combinations. One more role. You might be heterozygous. Oh no, that's hair color. My wife, when I married her, she was blonde. A typical Dutch blonde. All right. But now I know for sure that she's heterozygous. Because after we got married, within 4 or 5 years, because maybe this is inactive, right? For some reason this gene is not working. And of course you get the blue trait, right? But after 4 years, I don't know, the stress of being married to me, this gene became activated and her hair color changed to brown. Literally changed to brown. So I married a blonde, now I have a brunette color. I'm not complaining, all right? I like brunette. But sometimes it does happen. With eye color, it's not as straightforward. Sometimes people are born with 2 different eye colors, right? Brown and blue, you know. It's rare, it does happen. Some people suspect there's some kind of genetic abnormality, but we don't know for sure. Why is brown winning out? I want you to think of it as a factor. Okay? Go back to this. By the way, before I'm glad you guys answered this question. Is everybody clear about these test crosses?
Yeah.
Imagine you have a baby. He has an ugly parent. This baby is heterozygous. That's why you get the brown eye. All right? Well, I want you to think of these genes as factories. You have a dominant factory and you have a recessive factory. This one is producing blue pigmentation, this one is producing the brown pigmentation, right? Which one do you think is going to produce more? Because it's a bigger factory, right? More efficient. That's why what is happening is the brown is masking the effect of the blue is there. It's just being masked, but it's outproduced by the brown. So somehow, through some genetic abnormality, like my wife's case, this was inactive. And if this is inactive and this one is operating, you're blocked. And then you marry Masani and you go through stress, which is normal, and you end up with being married to me is stress. See how I am in the classroom? That's how I am at home. Even worse. I drive my wife nuts. I dress up like thieves and I break into the house to test her reaction. Every time. It's so easy. Grab the baby, panic room, hit the button, it's over. You're safe. Or go get the gun. Quick load. I have a revolver. Quick load. You're done. But one day she might shoot me.
So how would Mendel come up with the true breeding lineage? Like, how would he find out?
He just good question. How did Mendel came up with the true breeding lineage? People have been crossing stuff throughout the years, and they knew that these whites, every time you cross them, you always get white. And, and he even established his own lineage. You just cross over time. Yeah, went to the Home Depot and bought it. Okay? Yes?
So,
Yeah.
I got a question. You know how you said that they could go inactive, like your wife moves from blonde to brunette? Is it possible to go from brown hair to blonde hair?
From brown hair to brown hair?
Yeah.
It can go the other way around. It's like blonde to brown, or brown to brown. If something happens that switches off this gene and this gene is still active, it's possible. It's very rare. I mean, if it happens, probably 1 in, if I give guess, probably 10 million, maybe even more. It's rare. Even my wife's case is rare. But it did happen. Or she was lying to me. She was dyeing her hair. But I would have known. All right, somebody else has a question.
oh, I was just wondering then, okay, so then like your genes though, it also has nothing to do with like the sex, like which is more dominant, like if male genes are more dominant than female genes?
There is no male gene. Within the Y chromosome, there's a region called SRY, will determine if you have testes or ovaries. That's it. That's it. In terms of which genes are dominant, it's not the gene, but generally, technically speaking, it's the X chromosome. There is no male gene or female gene or which one is dominant. All right? Yeah, there's no dominance. Yes.
So you can do that Punnett square with the XY sometimes?
So what?
So you can do the Punnett square with the XY?
And we'll talk about that next week when we talk about X1 traits.
Alright.
But I want to get the foundation first, all right? But we will talk, you have to trace traits based on genes, okay? And we'll talk about that later, all right? So you're going to click on this. So when he did his test process, he came up with his law, Mendel's first law, okay? It's called the theory of segregation, or the law of segregation. And by the way, law of segregation, this is not Jim Crow law, okay? This is genetic segregation. Remember, up to this point, let me build it up if I can explain what this law is. I want to simplify it. Up to this point, people thought the traits are mixing, right? The traits are mixing. What Mendel showed us is, it's not mixing, because you're getting this separation, right? In order to get this, these two have to separate. These two have to separate and then individually get passed on. See what is happening? So what the law of segregation says, and I'm going to use modern language, you didn't know about this, but during meiosis, the diploid cell, they split, right? And you form 4 haploid cells. So what is happening is the gametes will inherit, or they will segregate, and you get a different allele in each one. All right? So best definition you can write this down. Alleles segregate from one another during the formation of gametes. Alleles segregate from one another. Alleles segregate from one another during the formation of. Now, the definition he used was a pair of, you don't have to write this down, a pair of characteristics, brown or blue eye color, for example, only can be represented in a gamete. In other words, you have this can be separated. It's a little bit more complicated. What he meant was that for any pair of characteristics, there is only one gene in a gamete, even though there are 2 genes for ordinary cells, right? An ordinary individual is diploid, 2N, right? But now you are having haploid. If this is the sperm, this is the egg, now you have haploid. What it's saying is these guys segregate and they end up in different alleles. Now, which one ends up with the other one? That's completely random. There's no law saying they don't have it to it. But they just segregate. To set the foundation rather than the mixing of the traits. Are you with me? Okay. I know, I'm running behind, I can't give you a break. All right, now, next one. And this one, you don't have to do Punnett squares. And we're going to do no Punnett square practice next week, too. So I want to make sure you get these. By the way, when it comes to calculating probabilities, don't panic. Each square is 25%, right? So if these 2 squares have dwarf, and I say what percentage is dwarf, you say 25, 50%, right? So you don't have to actually sit there and calculate. Or 1/4 and 1/4, and if you combine them, you get 1/2. So it's not that bad. Okay? There's no super duper calculations. So that was his law of segregation of characteristics. Character will segregate. His next experiment, and by the way, imagine, he did thousands of crosses, thousands of test crosses. He did it for dwarf, tall. He did it for wrinkle, non-wrinkle, purple flower, white flower, and he came up with these characteristics. All right, spent a lot of time on it. Then he did another series of crosses. He did dihybrid crosses. He did a dihybrid. Now, what is a dihybrid cross? Well, in a monohybrid cross, he was only crossing one trait. In a monohybrid cross, he was crossing only one trait. In a dihybrid cross, he was crossing for 2 traits at the same time. In his specific case, he was crossing purple and tall. So plants that are fruit-breeding ladies for purple and tall, and he was crossing them for the plants that were true breeding lineage for white and dwarf. So purple and tall being crossed with white and dwarf. That's it, white.
Yes.
White and, now, purple is dominant, he knew that, okay? Well, how did he knew that? Well, this is a parent generation. When he crossed them, guess what? All offsprings were purple and, so he knew purple was dominant, so capital P, tall is dominant, capital T's, right? P, P, T, T, dominant, right? And then what the white and dwarf want, lowercase t and lowercase t. He did those crosses. Then he said, if I am correct, all of these guys are purple and tall, all right? And if I do an F2 cross, in other words, if I take these F1s and I cross them with each other, I take the F1s and I cross them with each other, I should get a ratio of 9 to 3 to 3 to 1. 9 will be purple and tall. 3 will be purple and dwarf. 3 will be white and tall. And 1 will be what? White and dwarf. Right? That's what he said. And he did the cross. Yes?
I don't get it.
Okay. You get this part, right?
Yes.
Just the purple and tall.
Purple and tall, white and dwarf.
Often were all purple and tall, right?
Okay.
Then he hypothesized. He said, well, if this coding system is correct,
Right.
right, then if I cross these guys together, the F1s,
Okay.
I should get 9 to 3 to 3 to
2 1.
That's what you should get.
Okay.
Now, why is he getting this here? Remember, in the other one, when you had T, you separated them, right?
Right.
You separated them, right? Now, in this case, I'm going to use this as an example. P, P, T, T, right? You don't separate P, P, T, and T. These traits are joined together, right? They're on the same chromosome. So what you get is, you get this one and this one together. That's one. You get this one and this one together. That's one, right? You get this one and this one together. And, right? And if you apply the same thing to that one, lowercase p's and t's, you get all of them that happen to be what? 4 lowercases, p's and t's, right? Right?
So how did you get the 9 purple?
We'll get this is just this, all right?
Okay.
Guys are jumping in for one step at a time, all right?
Okay.
Now, he said, if I were to cross these guys, purple and tall, and purple and tall, right? I mean, white and dwarf, right? When you cross them. Now, we know these are the alleles, right? So if you combine all these alleles, all your offsprings will always be what?
Purple's the tall one.
We'll have a dominant allele, recessive allele, dominant allele, and a recessive allele, right? These are purple and tall, right? Right? Now, if I'm crossing these with each other, in other words, if I'm crossing this with this, if I'm crossing these with each other, right? He said he'll get 9 to 3 to 3 to 1, right? Well, let's do the alleles for this one first. Let's do the alleles. You have purple and tall that are heterozygous for both traits, right? This can go into one sperm or egg, right? This and this one can go in one sperm. You have a dominant purple allele and recessive T, right? This one and this one can go in there, so you have a recessive and you have a tall one, right? And this one combined, you have 2 recessive alleles, whiteness and dwarfness, right? So in other words, remember in the regular Punnett square, we did this, right? We put them like that, right? But how many do we have now? 1, 2, 3,
Four.
and So your square will be a 4x4 design. Your Punnett square will be,
4 by 4.
and by the way, on the exam, I'm not gonna have you do one of these 4x4s.
Okay, yeah.
So notice, these are all the alleles we get, right? So, purple and tall. And since you're crossing two of the same kind, right? This is what he's crossing, right? This side will be exactly the same, right? Right? So if we were to combine these together, two dominant T's and that, right? This with that, dominant T, lowercase t and T, right? Right?
Because I was switching. I thought it would go bigger than that.
Okay? So, we just finish this up, and then we'll go through this and look at their genome. By the way, he also did tri-hybrid crosses, and then you get 46 squared. Yeah, a lot of time. Oh, I didn't month. What did he do? Actually, he was very active in the community, too. So, it's amazing that he found the time. You have to stick with it. All right. Arms. I had this diagram, so transparency, but we don't have a transparency. What color is this going to be?
Purple, tall.
Right? I'm just going to say P, her, and tall. All right? What about this one?
Purple and tall.
Her and tall. What about this one?
Purple and tall.
Her and tall.
Purple and tall.
Purple and.
Oh.
Everybody sees that, right?
Yeah.
What about this one?
Purple and tall. White and tall. Purple and tall.
Tall. Wait.
White and tall.
What about this one?
Purple and tall. Purple and tall.
Tall. Purple and dwarf.
Purple and dwarf.
Purple and dwarf.
Purple and dwarf.
Purple and tall. Wait. Tall. Purple and tall.
Okay? Now, let's count the purple and tall. 1, 2, 3, 4, 5, 6, 7, 8. Any more? Right? Now, let's count white and tall. White and tall, 1. White and tall, 2. White and tall. Right? Now let's count the purple and dwarf. 1, 2, 3. Right? And then by golly, Miss Molly, the last one. White and dwarf. He got the 9 to 3 to 3 to 1. And by the way, he did thousands of Trex crosses for each one of these to determine if there's homozygous dominant for both of them or heterozygous for one. Insane. Okay? So on the exam, again, I'm not going to have you do Punnett square. I'm going to ask you conceptual questions. If I say true breeding lineage for purple and tall is crossed with a true breeding lineage for white and dwarf, you should say all the offsprings will always be purple and tall because they will be heterozygous for both traits, right? And if I cross two heterozygous ones with these traits with each other, you always get the 9 to 3 to 1 ratio, okay? I'm not going to get into his test process, but he also did a series of test crosses. And based on all that analysis, he came up with his second law. And the second law is the law of independent assortment. Law of independent what? Assortment.
Assortments. Assortment.
Technical definition that he gave, Mendel's second law, or the law of independent assortment, says, the law says that for two characteristics, the genes, and in parentheses I want you to put down alleles, are inherited independently. The genes are inherited what? Independently. Again, it says that for two characteristics, the genes, in parentheses, alleles, are inherited what? Independently. So, simply put, what it's saying is that when these genes assert themselves, right, when they're sorting themselves out, they sort themselves independently. There is no rule to it that the dominant one will go here, the recessive one will go here, and so forth. Same thing with your parents. When your parents and dad's chromosomes, right, you inherit a pair, right? During meiosis, remember, maternal chromosomes, do they line up on top, parental bottom? No, they can mix up, right? And when you're separating these chromosomes, you might get sperms that have mostly maternal traits in them, or you might get sperms that are mostly maternal, or you might get a combination of the two, right? And that's why you are not a carbon copy of your parents, and your parents are not a carbon copy of them, and so forth with you guys. And when you have your own offspring, they're not carbon copies. They'll have traits that might look like you, but there are people who have children that they look nothing like their parents, right? Have you seen that? You know, parents butt ugly, and then the kid is like gorgeous. Or the opposite, good-looking parents, and the baby is ugly as hell. That is nothing sadder than an ugly baby. Right? You don't have to lie to the parents. Oh, you're good-looking, and you go vomit. Right? So pretty much, look, simply put, look, you have two genes, right? Diploid? You have two genes, right? Diploid? You lost the genes. They just separate independently. They get packaged differently. Now, if you have multiple traits, you know, these two can sort out together, right? Or these two can sort out. How they get packaged is completely random. There's no rule, there's no pattern to it, okay? It's all random. And pretty much all this stuff laid the foundation of modern genetics, and then people did other crosses and so forth. All right? So independent assortment, hybrid crosses, we're not going to get into this. All right? Any questions so far? Again, I will never have you this. By the way, you did the purple crosses, purple tall with wrinkle seed versus purple tall with smooth seed. And so here, how many squares do we have? 16, right? With that one, you have 32 squares. All right? Insane. But, but, you know, he woke up in the morning, he prayed, and did his Abbey duties, and then he jumped on his research. Oh, he didn't have a wife or child. I don't think they could get married. So they didn't have many issues. There was no TV. You don't go out often, get eaten by the wolves. So, if I was back then, if I didn't get into college, I would have been a problem. So that's the only way you can pursue science. Right. Now, let's talk about dominance relations. Because up to this point, what we agreed on, that the dominant always wins out, true or false? True. But there are different cases. We have what we call incomplete dominance. We have codominance. Interaction between genes. And then last but not least, environmental effects. Now, I'm probably going to get to the first two. Again, on the exam, you don't have to memorize these. I will tell you if the relationship is not a straightforward Mendelian relationship, okay? I will tell you so you won't be confused. in an incomplete dominance, remember, we said tall always wins out, right? Tall always wins
Oh.
out. Well, in incomplete dominance, not the case. You get what we call intermediate, right? Snapdragons. How many of you seen a snapdragon tree? Not a tree, a flower?
Yeah.
Right? Probably somebody walked by and looked at it, and goes, snap, it looks like a dragon, so that's what the name stuck. Right? It looks like a snapdragon. Right? It looks like the head of a dragon. I don't know how they came up with it. If you are homozygous dominant, you're red. If you're homozygous recessive, you have a white flower. Okay? So if you're homozygous dominant, you are a red flower. Homozygous recessive, you are a
White.
flower. But if you were to cross a red flower with a white flower, you were to cross these, what do you get? Everything is going to be heterozygous, right? Everything will be heterozygous.
Zygous.
You would agree? Now, in a straightforward Mendelian sense, you expect all the flowers to be what color?
Red.
Red, right? Because R is dominant, right? But in this case, they're all going to be pink. They're all going to be what?
Recessive.
Something in between. That's what incomplete dominance is. The dominant allele is not truly winning out. So you get something in the middle. Everybody gets it? So imagine if Mendel's plant had incomplete dominance, would that 3-to-1 ratio or 9-to-3-to-1 ratio would stand? No, it would have screwed up his results, right? That's why he didn't pick the mice, because mice do have incomplete dominance with some trait that he wanted to study. It would have completely ruined it, all right? So, this incomplete dominance, these are the things that interfere with the Mendel's class. So everybody talk about incomplete dominance? Again, homozygous dominant red, homozygous recessive white, but if you're heterozygous, you happen to be pink. So on the exam I say, well, in a case of a plant that has incomplete dominance, these are the scenarios. What the offspring will be? Well, depending on the offspring. Now, what happens if we cross two pink plants together? If you're crossing the two pink plants together, what do you get?
Purple.
You get, this one is red, right? How about this one? These are heterozygous, are they going to be pink? And then, see that, now you're getting 1 to 2 to 1, right? You don't get that 2 to 1 ratio, right? And again, it's more. All right, moving on. Codominance happens in humans. Codominance occurs in a multi-allele system. So it's not like you only have two alleles, right? So far we've been talking about one dominant allele and one recessive allele, right? One dominant and one
Recessive.
In codominance, you have a multi-allele system. You can have, let's say, the blood type. You have a dominant allele that is represented by letter A for A. You have a dominant allele for B, right? And then you have a recessive allele that's a lowercase I. So you have two dominants, right? So if you happen to be this, or a dominant and a recessive, you're going to be a type A blood person, right? Dominant A, recessive. If you happen to be this, what type do you have? B, right? Both are dominant and dominant, recessive.